Thank you very much for the introduction, and thank you for the opportunity to have the Pindborg lecture for the Scandinavian Fellowship of Oral Pathology and Oral Medicine in 2021. It is a great honor for me to hold this lecture. The topic of the lecture is on tumor stroma, more precise tumor fibroblast interactions in oral squamous cell carcinoma, which has been the topic of my research for more than 20 years. Here it is the outline of the lecture. First, I will introduce you to the tumor microenvironment and the cell of interest of today's lecture, the fibroblasts. Then I would show you some indications that fibroblasts get activated quite early in the neoplastic process in the oral mucosa. And then I will go to the core of the lecture, which is on the mechanisms of tumor stroma interactions in oral squamous cell carcinoma. The focus of my lecture is not to convince you that the tumor stroma is important, but more to go into this mechanism of this interaction and how we can use the knowledge derived from this mechanism to target tumors for a better treatment of cancer patients. I would like to start pointing the attention to a study from 1960s. Today, this study will be ethically unacceptable. What the researchers did at that time, they took epithelial neoplastic islands from tumors of the patients and they transplanted in the back of the same patients, either taking with epithelial islands the stroma or without the stroma. What they found out was that if they would take together with epithelial islands the stroma, they will manage to get a new tumor in the new site. While if they will not take the stroma, then the tumor cells will die, and within 3 weeks it will be no tumor in the new site. This has been one of the earliest indications that tumor stroma is important, and also the way the host reacts to the transformed cells is important. Another very interesting study is the study which has been performed by Folkman and Kaluri. In this study, they have investigated the breast of the women that have died by accidents, in other words, by other causes than cancer, and they could identify in women between 40 to 50 years of age, almost 1/3 of the females have transformed epithelial cells in the breast. Compared to the prevalence of the breast cancer in the same age group, which is approximately 1%, this is a huge difference. So many of us carry also, the study has been performed on thyroidea, and they could find that in patients between 50 to 70 years, almost everyone had transformed cells, but the clinical prevalence is much lower. So we might carry these cells, the transformed cells, epithelial cells, but they don't develop into a clinical cancer if the host doesn't allow it. And Folkman has been interested in vascularization, which is one component of the tumor microenvironment. I am interested in fibroblasts and how they interact with the tumor cells and also with other components of the tumor like the blood vessels. But in the tumor microenvironment, we find also nerves, we find fat cells, we find also inflammatory cells. So depending on which is your focus, one can look at the tumor cells, one can look at the fibroblasts, the blood vessels, or your cell of interest. Today's topic is about the fibroblasts. What is a fibroblast? Despite of the interest in this cell and the interactions with tumors and its relevance for the tumor progression, we still have a pretty poor definition of this cell type. It is usually defined by a combination of morphology, spatial location, and lack of other lineage markers. Here I took some pictures from a normal human oral mucosa. You could see these are the cells which are elongated. Here they are depicted with vimentin. And we see them when we take them in culture. They have this fusiform shape and they are quite motile and they express usually vimentin. They have a very important function for our tissues. They are very important for the formation of basement membrane, for the remodeling and synthesis of extracellular matrix. They synthesize many soluble growth factors. They regulate epithelial differentiation. They regulate also inflammation, and they are very important for the wound healing because they get activated into myofibroblasts that migrate into the wound. They contract the edges of the wound and synthesize new extracellular matrix. The myofibroblasts in the wound healing, they will disappear when the connective tissue is restored. However, there are differences between fibroblasts in different sites of the body in terms of how do they get activated and how they contribute to the wound healing. So for long it has been observed that if you have a wound into a mucosa, after 1 week you have no scar, while in the skin there is a scar that is persistent, could be for all your life. So very interesting early work from the group of Phil Stevens from Cardiff have shown that the oral fibroblasts are very different from the skin fibroblasts. So they produce increased amounts of hepatocyte growth factor or scatter factor, and they also stimulate much more endothelial cells, in addition to producing more MMP2, being more motile, and contracting more the collagen lattices. Why they are so different, we are still investigating that, but what we know is some of the oral fibroblasts, they have a neural crest of origin, and this has been demonstrated by several studies. They also express some neural markers. Of interest is also that oral fibroblasts seem to be able to differentiate in cells that can give rise to bone proteins, to cartilage, and to fat. However, we know that the oral mucosa differs in oral cavity, and so the fibroblasts also differ. The major difference seems to be between the periodontal fibroblasts and the buccal fibroblasts. Again, they are also very different from the skin fibroblasts, and this is a study from 2003 that have investigated that. This has been some information about normal fibroblasts. As I mentioned, we now have a massive amount of evidence that the fibroblasts get activated with the neoplastic process. And here I took the data from the protein atlas, and here you could see that head and neck tumors, they are some of the tumors that have pretty abundant tumor stroma compared to other cancer types. And of course, they are quite heterogeneous. We know that in terms of the genetic makeup of the cells, but also in terms of the tumor stroma. So some have an abundant tumor stroma, some have a less abundant tumor stroma. Here is depicted in two different patients with alpha smooth muscle actin, which is a marker of the myofibroblast. So then we deal again with the same question: what is then a cancer-associated fibroblast? It is a fibroblast that surrounds the tumor islands, of course. So again, the definition is also by morphology, by spatial location, and lack of lineage markers. This is a good review for some years ago, but still stands true, by De Wever, who looked at myofibroblasts and cancer-associated fibroblasts and the markers that they express. The problem is that these fibroblasts, they express markers which are expressed in a plethora of other cell types, as mentioned here. The only ones that seem more specific are the alpha smooth muscle actin. That's why a lot of attention has been to the myofibroblasts that express the alpha smooth muscle actin, and also the expression of prolyl 4 hydroxylase. I would like also to draw attention to a marker which at that time was pretty new, the integrin alpha 11, which is an interesting marker because it looks like it's exclusively expressed by cancer-associated fibroblasts, and it's not expressed in any other tissues, a part of the periodontal tissue. Some of the earliest indications that we have activation of fibroblasts into myofibroblasts by oral squamous cell carcinomas come from a paper from 2004 in British Journal of Cancer, the paper of Lewis, which shows that if you isolate fibroblasts and you expose them to conditioned medium from different oral squamous cell carcinoma cell lines, you get them activated into myofibroblasts, here depicted in green with the alpha smooth muscle actin visualized. And also this activation seemed to be very similar to the activation of fibroblasts by the growth factor called TGF-beta1. They could show that the fibroblasts stimulated to express alpha smooth muscle actin, they also synthesize high amounts of hepatocyte growth factor. This seemed to be a factor, a growth factor that appears again and again in the topic of fibroblasts, either normal or cancer-associated fibroblasts. And these fibroblasts, once activated, they were able to stimulate the invasion of the oral squamous cell carcinoma. After that, alpha smooth muscle actin has been investigated in patient material. And here there are two studies published almost at the same time, one from the lab of Gareth Thomas, one from the lab of Tula Salo. One looks at oral squamous cell carcinoma, the other one looks at tongue, mobile tongue carcinoma. They both show that in some cases there is increased expression of alpha smooth muscle actin, and this one is correlating, higher expression of alpha smooth muscle actin is correlating with a poorer prognosis. However, they both show that there is a variation. There are some tumors which are negative, and there are also some tumors which has an intermediate phenotype, and there are some tumors that have a really rich myofibroblastic stroma. I would now move to discuss a bit when the fibroblasts get activated. And as I mentioned, we still struggle to identify good markers for cancer-associated fibroblasts or activated fibroblasts, but we have one marker, which is the alpha smooth muscle actin for activation of fibroblasts into myofibroblasts. And this has been used in dysplasias to look for early activation of fibroblasts in these lesions. This is a paper looking at low-risk oral dysplasia compared to high-risk oral dysplasia, and they find an association between expression of alpha smooth muscle actin and the degree of dysplasia. We looked also with two master students, and here you could see oral mucosa from a patient that presents multifocal areas with p53 mutated cells, and you see here a skip lesion. And then, very interestingly, you could see the inflammatory infiltrate under exactly just these areas with p53 mutated cells. And if you do an alpha smooth muscle actin staining, you could see not only the blood vessels positive, but also you could see single elongated fibroblasts, which are situated right under the p53 mutated clusters. So we found an association between the two stainings, the positive staining for alpha smooth muscle actin and the positive staining for p53, which indicates that there is a mutation in the p53 tumor suppressor gene in these areas. If we look at the expression of alpha smooth muscle actin now into isolated fibroblasts from under dysplastic lesions in oral mucosa. This is a study that we have performed for some years ago. And then actually we could see that the expression of smooth muscle actin varies quite a lot in these isolated fibroblasts. And we couldn't see any pattern in the fibroblasts derived from normal mucosa, derived from under dysplastic lesions, or from oral squamous cell carcinoma. However, when we did the transcriptomic analysis, we could see that the fibroblasts from under the dysplastic lesions, they clustered separately away from the normal fibroblasts and quite localized compared to the more heterogeneous cancer-associated fibroblast population. Interestingly, when we put them in culture with a cell line, which has been defined as dysplastic because at that time didn't form tumors in nude mice, we could see that they could stimulate some invasion of cancer cells. I would like now to present very briefly the models I'm using, which are the 3D organotypic models, because they are very useful for the tumor-stroma interactions studies. So what we do, we get a small biopsy. Usually we get it for normal mucosa from the clinics in Bergen town after the extraction of wisdom tooth and after informed consent. And we think simplistic. It's a model, so we think of the epithelium being composed of epithelial cells and fibroblasts. We isolate these two layers using this space. We propagate the cells separately in their lineage-specific medium, the epithelial cells and the fibroblasts, and then we reassemble them together in the so-called raft or organotypic culture. Here is an image of a native mucosa, and here is an image of cultivated normal mucosa, which needs to be lifted at the air-liquid interface after a few days, because this will induce proper differentiation of the epithelium. So this is the steps that we are using in culture. As I mentioned, using this space overnight, we could separate the epithelium from the connective tissue. We then propagate the cells in monoculture. We then seed the fibroblast into collagen on ice. It's liquid when we put it in the incubator at 36 degrees, and also into a physiological pH. It gets jelly, and then we seed the keratinocytes on top. We let the cells to grow together for a few days, and then we lift them, and we obtain this small artificial mucosa. In this way, we can generate various models, because we can either alter the epithelium compartment or the mesenchymal compartment. And during the presentation, you will see many results generated using these models. So here, for example, is the original lesion of a oral squamous cell carcinoma, and how we use the cells from this patient to generate artificial small tumor. So using these models, we have analyzed the effect that the dysplastic or the fibroblasts from under dysplastic lesions can have on this dysplastic cell line. And you see, while the normal fibroblasts seem to restrain the invasion of these cells, the dysplastic fibroblasts, they seem to stimulate or to allow these cells to invade. However, this invasion was not as pronounced as if you would have cancer-associated fibroblasts, which were stimulating very much the depth of the invasion. So we have indications that the activation of fibroblasts goes pretty early. This comes mainly from investigating myofibroblasts. Could be that we have other types of fibroblasts that get activated early, and there are indications that lysyl oxidase like 4 could be also one enzyme that gets overexpressed in the early activated fibroblasts. However, I will move now towards the clinical relevance of this. So, okay, there is evidence that myofibroblasts are present in early transformed epithelial lesions. However, does this have any importance for the further progression to cancer or for the life of the patient? And a very good systematic review from the Coletta and Salo shows that actually there is no evidence, no such evidence that the presence of myofibroblasts in lesions with malignant potential can predict the malignant transformation. We move now to the core of the lecture, which is on the mechanisms of tumor fibroblast interactions in oral squamous cell carcinoma. Here is an overall graphical abstract of these interactions. So we have here the cancer-associated fibroblasts, and it is now revealing that they are quite heterogeneous. So here with green are depicted a subpopulation that seem to have tumor restraining abilities, while the majorities of the studies actually, and a lot of the focus have been on the fibroblasts, which are here depicted in brown, which have been shown to have tumor-promoting interactions. So I would like to start looking a bit into this area, which is less explored. And this is a study from Patel et al. 2018. They have isolated fibroblasts from oral squamous cell carcinoma, and they looked at the expression of alpha-smooth muscle actin. So in other words, the percentage of myofibroblasts in this population. So this is an experimental model. However, they could see that the fibroblasts they have isolated, similar way as we have seen, is quite heterogeneous in terms of expression of alpha-smooth muscle actin. Interestingly, when they did transcriptomics, and here you see the dendrogram, when it was an unsupervised clustering of the 1,000 most altered probes, they could cluster the fibroblasts in two clusters. They named them C1 and C2. And interestingly, they seem to be correlating with the clustering according to the alpha-smooth muscle actin. So the C1, they were the fibroblasts that had a low expression of alpha-smooth muscle actin, while the C2, they had high expression of alpha-smooth muscle actin. When they looked at the interactions with cancer cells, they made at least two very interesting observations. One was that the CA1 type of cancer-associated fibroblasts, they could stimulate the proliferation of cancer cells, different cell lines. However, they didn't seem to support the expression of ILDH1, which is a marker of stem-like cancer cells. Interestingly, the C2 type of CAFs, these are the myofibroblasts, they have high percentage of myofibroblasts. They seem not to support so much the proliferation or no significant difference in the proliferation of the cancer cells. However, it was a significant increase in the expression of ILDH1, meaning that these fibroblasts were able to support the stemness or the stem-like properties of tumor cells. So they put up these very interesting models correlating it with data from patient material in which they could see a correlation between alpha-smooth muscle actin expression in the stroma and the expression of stem cell marker ILDH1 and an inverse correlation with the proliferation marker Ki67. So this is one of the first indications that we have a tumor suppressive subpopulation of fibroblasts in the oral squamous cell carcinoma. It seems that it acts through BMP4, which is suppressing the stem-like cancer cell phenotype. Moving further, even one more layer of complexity into the heterogeneity of cancer-associated fibroblasts, and also with some relevance for the tumor suppressive effects of CAFs, is the study of Melon from 2017 from the group of Gareth Thomas, which looked at the alpha-smooth muscle positive cancer-associated fibroblasts. And they identified two subpopulations within this population of myofibroblasts. One seemed to express P16, which is a marker of senescence, while the other ones didn't. Here you could see that some fibroblasts that express alpha-smooth muscle actin, they also express P16, while some others, they don't. When they did transcriptomics using TGF-beta-1 stimulation, which would be the treatment that would increase the myofibroblast phenotype compared to the irradiation phenotype, which will be the phenotype, the senescent fibroblast phenotype, they found out that there were some overlapping genes. However, they were much less compared to the genes that differed between these two types of fibroblasts. So the genes that were similar, they belong mainly to the motility pathways, while the genes that were separated in the TGF-beta stimulated fibroblasts, they belong mainly to the matrisome core. So these are the genes that are involved in the synthesis of extracellular matrix proteins, while the other ones, the irradiated phenotype, had the senescence signature. Furthermore, they looked at how this would impact the oral squamous cell carcinoma invasion. And both in the in vivo culture, on models, on in vivo and in vitro models, they could show that the TGF-beta1 stimulated fibroblasts, they could synthesize collagen that was also very well organized, while the irradiated fibroblasts, the more senescent fibroblasts, they did not express so much alpha-smooth muscle actin, and also they had less collagen deposition, and the collagen was not so well organized as the TGF-beta1 stimulated fibroblasts. And this had an impact on the invasion of cancer cells, as you could see here. They moved also to patient material, and they could show that the presence of the fibrogenic fibroblast, as indicated by the type of collagen and the organization of the collagen fibrils, was predictive for the progression and the prognosis of head and neck squamous cell carcinoma, which included also oral squamous cell carcinoma. These are, by my knowledge, the studies that show that there is a subpopulation of fibroblasts which might have some tumor restraining interactions. The evidence pointing towards tumor promoting interactions between fibroblasts and epithelial cells and tumor cells is much more overwhelming. So I picked some studies looking at mainly the support the CAFs give for proliferation and survival, for invasion and metastasis, also studies that look at the metabolic fuel that CAFs provide to cancer cells. And I will also very briefly touch upon the interactions that cancer-associated fibroblasts have with other cells in the tumor microenvironment, but which has an impact on the tumor. So it's also a tumor-promoting effect, which would be angiogenesis and immune cell evasion. Amani Hamza, a PhD student in my lab, was very much interested in the plasticity of cancer stem cells. But surprisingly, her attention was quickly drawn to the cells that were more differentiated, the CD44 low cancer cells, which actually reacted much more to the presence of cancer-associated fibroblasts. They became more clonogenic, and they also survived more and were propagated more in culture. These studies are performed on primary cells isolated from patients, and we haven't sorted the cancer-associated fibroblasts in subtypes. So this is a bulk of cancer-associated fibroblasts and the bulk of cancer cells that has been sorted for CD44 or for rapid addition, for increasing in stem cell or depleting in stem cell population. So the response of different patients was various, but there is a trend in which the CAF stimulate the clonogenic ability of CD44 low cells. Very few patients could develop spheroids when we put the cells, either the CD44 high, which are enriched for stem cell-like tumor cells, and for CD44 low, almost none of the patients form spheres. However, when we added cancer-associated fibroblasts, some of the patients form spheres with even CD44 low cells. And this we could observe it in so-called dysplastic cell lines, not fully transformed. This effect, we could observe it in cell lines, but somehow not so obvious. When we did animal models, we could also see that tumor take was increased by co-injecting CD44 low cells together with cancer-associated fibroblasts, and the tumor take was pretty similar to the tumor take of CD44 high cells. The tumors formed by the CD44 low, when they were formed, they were very small and they had a pushing border type of invasion. When we added CAFs, we could see that the pattern of invasion has changed. We could see trails of cancer cells invading into the tongue, and also we could see metastasis. Amani was wondering if this is due to direct effects, meaning direct contact, or if it is due to conditioned medium effects, and she identified that TGF-beta1 seemed to be the essential growth factor that is mediating this effect. And very interestingly, the effect on increasing the clonogenicity has been much higher on the differentiating population of the oral squamous cell carcinoma compared to the less differentiated. And we could see that these cells got activated compared to the CD44 high cells. Of interest, it was also that she investigated the TGF-beta receptors and their activation, and it looked like there is a difference in the way these receptors are expressed between the more differentiated cell type and the more stem-like tumor. So this has been some piece of evidence for the increase in survival that cancer-associated fibroblasts have. And this is interesting because when the field of cancer stem cells was blossoming, the theory was put like, if we manage to eradicate the tumor stem cells, then we will eradicate the tumors. However, these results show that if you manage to get rid of the cancer stem cells, the more differentiated cells are also very plastic. So the plasticity goes both ways, from stem to non-stem, and also from non-stem more differentiated to stem cells, and they can repopulate with stem cells a tumor. Now I would go towards more invasion and metastasis studies. And this is a study from our group. It's the same study that I have shown you some results on the cancer, on fibroblasts derived from dysplasia. So here, what we have observed when we cultured in tissue fibroblasts isolated from cancer patients with different cell lines, we have observed a difference in the pattern of invasion. Some fibroblasts would support a very deep invasion in these 3D organotypic cultures, while some will support some invasion much deeper than with normal fibroblasts, but not as deep as with the other one. So it was a variation. We did transcriptomic analysis, and here I have showed to you this unsupervised clustering when showing the fibroblasts from under dysplastic lesions that clustered separately from the normal fibroblasts. They were quite grouped. However, the cancer-associated fibroblasts were much more heterogeneous, but we could group them in two different groups that actually mirror their behavior in vitro. So we have some fibroblasts, the cluster that is closer to the normal fibroblast, and the cluster which is much more divergent. So we call them CAF-N and CAF-D. At that time, when we performed this study, was published a very interesting paper by Gaggioli, which showed that fibroblasts, here marked with red, are actually the cells that are leading, that are making tunnels in the collagen gels and are leading the invasion of the cancer cells, here depicted with the green. So here is a movie showing a red fibroblast moving ahead and followed by a green cancer cell. Interestingly, they have followed this study, and I will very briefly show this because I think it's fascinating. They have looked, this is data from fibroblasts isolated from vulva squamous cell carcinoma, but it has relevance for oral squamous cell carcinoma as well. So what they have investigated here was the traction force of the fibroblasts nearby the spheroid of the cancer cell. So here in green is the spheroid of cancer cell, and with red are the fibroblasts, cancer-associated fibroblasts. And they could observe that initially, before contacting the spheroid, some of the fibroblasts, they had quite even distribution of the traction force. However, when it was in contact with the spheroid, it started to pull the cancer cells, and you could see here by this vector the tracting force. So here it's a video showing how the red fibroblasts are basically dragging the cancer cells to invade. And there seemed to be a heterogeneity among the fibroblasts. So some seem to have this ability, as you could see here, to drag the cancer cell, while some others are coming, exploring, and move away, and they call them this solitary or loner, and this a leader fibroblast. They even went into the mechanisms, and they have shown that it is due to the interactions between N-cadherin and E-cadherin. Here the E-cadherin is in red and the N-cadherin is in green. So now it's the other way around. The cancer cells are red and the fibroblasts are green. And here you could see how the fibroblast is having these very close contacts here. And they have shown evidence that if you neutralize this interaction, then the fibroblasts don't manage to drag the cells anymore. And interestingly, in this paper, which is a collaboration with Eric Sahai, they show that in normal mucosa, you don't have these kind of interactions, but you have them in squamous cell carcinoma. And this is data from oral squamous cell carcinoma. Now, going back to our study. So have in mind that at that time, while we were investigating and trying to understand and to corroborate the transcriptomics data with the motility data and the invasion data that we found in vitro, came the study of Gargioli. So then came the idea that we should look at the migratory properties of fibroblasts. And what we found out was that, yes, indeed, the cuff ends, those closer to normal fibroblasts, they had a higher ability to migrate, and also they were more prone to get activated by TGF-beta. So they both migrated longer, and it was a more directionate movement, not all around. So interestingly, one of the genes that came on top of our transcriptomics data was hyaluronan synthase, and when we knocked that down with shRNA, we could see that actually that had a major impact on the invasion of the adjacent cancer cells. I would like to draw a bit attention of what do we mean by desmoplastic stroma in oral squamous cell carcinoma, because at least in research, a lot of attention has been drawn by the fibrillary deposition of extracellular matrix as a definition of desmoplastic stroma. However, this data comes mainly from breast and pancreatic adenocarcinoma, where indeed you could see here in blue is depicted the collagen with trichomescens, while here with the Alcian blue we see hyaluronic acid and glucosaminoglycans. But in squamous cell carcinomas, and I'm talking both about oral and vulva, what we see is that, yes, we see this fibrillary deposition, but mainly in the tumor center, while at the tumor front, as you could see in this case, for example, we don't see so much deposition of collagen, but we see deposition of ground substance. So it is a new stroma, it is a desmoplastic stroma. Here we could see that it's actually full of hyaluronic acid. So moving more in understanding how the fibroblasts interact with the stroma, with the tumor cells and their impact on the tumor cells, we followed this study. So having the same fibroblasts, and I have to mention these are grown in three-dimensional, and we have shown that the gene expression and the microRNA expression changes completely between monolayers and 3D cultures. Here we followed this transcriptomic study with the microRNA expression study. And interestingly, what we have seen, this is the list of genes that were upregulated in calves, and you see here the hyaluronic acid synthase, and you see also the integrin alpha 11 that I mentioned as a new coming marker for cancer-associated fibroblasts. What we have noticed in the microRNA expression, unfortunately, we couldn't define subtypes or subclusters of cancer-associated fibroblasts in this data. However, several of the microRNA that came on top dysregulated in the cancer-associated fibroblasts, they regulate integrin alpha 11. So while most of the studies looking at the tumor stroma interactions, they look at the proteins, they look at the proteomics, transcriptomics, there is now data coming that the microRNA and their regulation in the fibroblast in the tumor stroma seem to be important as well for the invasion and tumor progression in oral squamous cell carcinoma. This is our own study, which is now in revision, is the work of Saroj. And what he did, he used inhibitors and mimics of the microRNA 204, which by in silico has been shown to target integrin alpha 11. And actually, he could show that if he uses mimics of 204, so by increasing the expression of 204 in the fibroblasts, he could actually decrease the invasion in these 3D organotypic models, while when he used inhibitors, he could increase the invasion. And this went through the modulation of the migration ability, again, of the fibroblasts that was related also with integrin alpha 11 and with a CAF phenotype. So going back to our transcriptomics data, the first subtype of fibroblast that we have identified has as main mechanism of interaction with cancer cells through their motility and probably making the tunnels that the cancer cells are following. However, we were wondering what would be the mechanism of invasion in the other fibroblasts, this more divergent fibroblasts that don't have this high motility. And what drew our attention was a TGF-beta signature. And these fibroblasts, the CAFTs, they synthesize huge amounts of TGF-beta-1. So we showed that the mechanism through which they stimulated the invasion of oral squamous cell carcinoma was through induction of EMT, epithelial to mesenchymal transition in the adjacent cells, and this could be inhibited by addition of small molecule inhibitors for TGF-beta-1. So to summarize, we have shown both transcriptomics and functional data for two subtypes of fibroblasts. We call them CAFN, which are more migratory. They synthesize high amounts of hyaluronic acid. They support aggressive invasion. And they're quite responsive to TGF-beta and most likely form tracks in the stroma for cancer cells to follow. However, there seems to be another population that has a different mechanism of supporting invasion of oral squamous cell carcinoma cells, which are more stationary, but they synthesize TGF-beta, but themselves they are refractory to TGF-beta. And what we actually put forward, we analyzed several strains we had in the lab, is that it's not the new phenotype that is occurring into the tumor stroma. This types, they exist also in the normal stroma. However, the balance between these two varies. So either you have a more migratory phenotype or a more TGF-beta-1 synthesizing phenotype in various proportions in the cancer. Very interesting, another study showing that there are differences in tumors according to how the cancer-associated fibroblasts synthesize TGF-beta-1 comes from the lab of Prime and Ken Parkinson. And then they showed here that the fibroblasts from genetically stable squamous cell carcinoma, they synthesize not so much TGF-beta-1, but the fibroblasts isolated from the genetically unstable oral squamous cell carcinomas, they synthesize high amounts of TGF-beta-1, and this was associated with a phenotype related to senescence, and it had an impact on decreasing the E-cadherin, so again inducing EMT, corroborating actually with our results. So here they very nicely put up a model in which you have the tumors with a wild type P53, so they are stable, and then there is no increase in ROS and probably not so much TGF-beta-1 synthesis, but could be other changes into the fibroblast. However, in the oral squamous cell carcinomas, which are genetically unstable, these cells produce much more ROS, and they activate the fibroblasts. They activate in the sense that they induce them into senescence, which is associated with increase in TGF-beta-1, and this in turn has an effect on their invasion, as shown here. I will move now, related to the paper I just showed with the ROS production, to the interactions at the metabolic level. But before that, I just want to mention that most of the studies have focused on the interactions using soluble factors and proteins and growth factors, but there are now more and more studies which are very nice reviewed in this review in oral oncology, in which the fibroblasts seem to alterate the gene expression and the invasion motility of cancer cells through synthesis of extracellular vesicles that contain a range of microRNAs that can alter migration, invasion, and proliferation and survival of cancer cells. I will not go into those studies, but I will go now towards metabolic fuel because I have some new data I want to show you, and just to introduce you to the reverse Warburg effect. We are all familiar with the Warburg's original observation that the tumors show a metabolic shift towards aerobic glycolysis. However, Lisanti has observed that that's true when you have the cancer cells growing alone in culture, but however, when you put them together with the fibroblasts, actually the cancer cells induce the fibroblasts into glycolysis, and they use them as an energy fabric. And as a proof for the energy transfer between fibroblasts to cancer cells, he used the MCT markers, which are the MCT4 is a transporter of lactic acid outside, from inside outside in the tumor stroma, and MCT1 is a transporter inside the cell. So the cancer cells will have the transporter bringing in the lactic acid, and the cancer-associated fibroblasts will have the receptor that will bring out lactic acid. So evidence for the presence of this mechanism in oral squamous cell carcinoma has been provided by Jensen and a study in which Eric Dabelstein was involved in 2015, and they show that while you don't have expression of MCT4 in the normal oral mucosa, you have some expression in cancers, but it varied. And interestingly, they did serial sections for MCT1 and MCT4, and they showed that they were mutually exclusive. So the transporter in was in cancer cells, while the transporter out was in fibroblasts. However, not all the fibroblasts have expressed this. And even more interestingly, it looks like it was not a colocalization with the myofibroblasts. So their conclusion was that yes, a reverse Warburg metabolism might be in oral squamous cell carcinoma. They have some evidence for it, but they couldn't find enough evidence that the myofibroblasts are involved. We followed up that studies, and in our co-culture models, we labeled the normal oral fibroblasts with red. The mitochondria are red in these fibroblasts, and we have in green the cancer cells or the normal oral keratinocytes. And when we put them together, interestingly, we have seen not only, and we have proven in 2 papers, that there is energy metabolism transfer and lactic acid transfer, but in addition, we could see mitochondrial transfer. So it was a mitochondrial transfer. Now you could see in the green cells red mitochondria, or mitochondria that are fused, the red and the green, and they are yellow. This was pretty preferential just for the interactions between fibroblasts and oral squamous cell carcinoma cells. We haven't seen it in the normal oral keratinocytes. We have seen some reverse transfer from cancer cells to fibroblasts, but it was mainly a unidirectional transfer. And this has been very important for example, how the oral squamous cell carcinoma cells become more resistant to metformin treatment in the presence of fibroblasts. So in addition to proving a reverse Warburg effect in oral squamous cell carcinoma in an experimental model, we could show in addition what we call the modified reverse Warburg effect, in which we could observe also mitochondrial transfer from fibroblasts to the cancer cells. And now we are investigating the consequences of this transfer. Interestingly, this is a mechanism that has been described to occur in the injured brain first, but also it looks like this horizontal transfer occurs between astrocytes and neurons, and this is how neurons that need an enormous amount of energy to function, they get that energy also from the supportive cells in the nervous system. So this is a very interesting mechanism. If the cancer cells also hijack the mechanism that is physiologically present into the brain, it gets activated in cancer. So I took you through the direct interactions between the cancer-associated fibroblasts and cancer cells, showing some evidence for either supportive or restraining tumor stroma interactions. But cancer-associated fibroblasts, they interact also with other cells into the tumor microenvironment. So for example, they can interact with the endothelial cells. And here we show, as I mentioned, since alpha-11 integrin came up on top of our genes upregulated in cancer-associated fibroblasts from our early transcriptomic study, we have focused and tried to understand the role of integrin alpha-11 in oral squamous cell carcinoma. And interestingly, we found out that yes, indeed, it has a role for tumor progression. And here you could see these are mice that have been injected with cancer cells. together either with cancer-associated fibroblasts with expression of alpha-11 integrin, or that have been knocked down for the expression of alpha-11 integrin. And you could see that if you knock it down, you have smaller tumors, as shown here. And interestingly, what we could see was that these tumors, they were almost devoid of blood vessels, indicating that there is an interaction between this cancer-associated fibroblast in stimulating the vascularization, and this goes through integrin alpha-11. And we showed it experimentally that this is the case. Another very important interactions that fibroblasts have with other cells in the tumor microenvironment is with immune cells. So here is an overview over the interactions of cancer-associated fibroblasts with different immune cell types. So there is evidence that they interact with monocytes, and they direct them towards one type of another of macrophage. Also, they seem to have an impact on stimulating tumor suppressive T cells and suppressing the effect of T cytotoxic cells. And also, they seem to have an impact on the osteoclast. And I would like to mention a very interesting study that has been published in British Journal of Cancer, in which first it was an observation that us pathologists might make while looking at the slides in the microscope, and it was that actually the lesions that were invading the bone, it was not the cancer cells that were very close to the bone resorption areas, but it was an abundant stroma. And this abundant stroma expressed some markers that are important for the osteoclastogenesis. And using also experimental models, this group have shown that the fibroblasts were able to stimulate osteoclastogenesis in vitro, suggesting that the fibroblasts are actually the key component involved into the stimulation of bone resorption, and they suggest that the cancer-associated fibroblasts are actually leading the bone invasion in oral squamous cell carcinoma. I show you only one study that shows experimental evidence that the cancer-associated fibroblasts are able to suppress T cell proliferation, that they are able to induce T cell apoptosis and a phenotype of T regulatory or FOXP3 positive cells, which is pro-tumor. In addition, they show that the expression in cancer-associated fibroblasts of ligands for PD-1 and PD-2, they are upregulated in cancer-associated fibroblasts compared to normal fibroblasts, and this has putative negative effects for the immune function. And later on, I will show you why this is important. I hope the information I gathered here hasn't been too overwhelming. I tried to take you through seminal papers, studies showing both tumor restraining and tumor promoting interactions. And this is the summary of the knowledge that we have today, that the tumor stroma interactions in oral squamous cell carcinoma, mainly the tumor fibroblast interactions, are very important for cancer cell growth, for survival, for motility and invasion. We see also metabolic coupling, and we are investigating now how this affects drug response. And also cancer-associated fibroblasts seem to have a critical influence on immune surveillance. Now I will present just few points on how we can use this knowledge, how relevant is the knowledge derived from our studies on tumor stroma interactions for further work for improving the patient treatment. And one obvious reflection is that we have to move to use more relevant models. So we should move from monoculture or monocellular models to multicellular so-called interaction models. And we could use both 2D or 3D. There is a majority, there is a plethora of different models, and very good work from, for example, Tulasalo's lab shows that one can use these small organoids or different spheroids as avatars for the tumor progression, and you could expand them in culture, you could treat them with different drugs, and then you could depict the drugs that are efficient in that particular patient. Also, we could construct now in vivo models in which we combine, as I have shown the study on integrin alpha 11 and angiogenesis. I personally prefer in vitro avatars and in vitro work, but when necessary, for example, when looking at, for example, metastasis, and here you could see cancer cells in red and fibroblasts in brown, and you could see here a blood vessel in which there is a clump of cancer cells and fibroblasts that goes into the blood and then ends up into the lung. So these models are also very useful, but we have to use multicellular models because we need to develop better and more efficient ways for treating oral squamous cell carcinoma. So the knowledge derived from these tumor-stroma interactions will be very useful for patient stratification. But if we stratify the patient, we have to have a treatment available for the patients, for the different subclasses of patients. And that's why this all the work on tumor-stroma interactions and the mechanisms and identifying new molecules that can be targeted is very important. And we are moving now from the first thought that if we would just eradicate the cancer-associated fibroblasts in a tumor, then we will treat the tumors. That seems not to be the case. Most likely, we can modulate the cancer-associated fibroblasts and the molecules they synthesize, and also we could use maybe precursors of cancer-associated fibroblasts, the mesenchymal stem cells, for drug delivery into the tumor. So there are many different aspects in which tumor-stroma interactions are important. I would like to end with one study. It's not from oral squamous cell carcinoma, but it is relevant. So in this study, which comes from Gareth Thomas' lab again, they put up a model in which they could target an activated stroma, the cancer-associated fibroblasts that seem to mediate exclusion of cytotoxic T cells by using the ROS inhibition, the inhibition of the enzyme NOX4, which is involved in ROS. And they seem to get pretty good results by doing that. With that study, I conclude my presentation for today. And I would like to thank very much to my research group, which is a hard-working, hard-party research group, to my collaboration, of course, to the clinicians and to the patients and volunteers that donate us their tissue, because we are dependent on them. And recently, actually, we got ethical approval to isolate the cells from autopsies, and we managed to isolate live cells for 6 days post-mortem, and that's a fantastic source. So thank you to all patients donating us their valuable material, and also thank you to the financing bodies, and thank you for your attention.